I like to tell you indeed is something about the immunology of viral hepatitis, and I'm going to focus on hepatitis B, leave out the HIV a little bit because this whole area is confusing enough. And so what I'd like to show you first is to show you the two examples of a very successful approach. One approach, of course, and that also is very important from the immunological point of view, is vaccinations. And so this is a study recently published from Taiwan, in which they had the follow-up data from a 30-year of very effective vaccinations in Taiwan. And you can appreciate here that at the start of the whole vaccination program, about 10% of the patients were HBs antigen positive, and after 30 years of effective vaccination, only 0.5 of the patients, or of the individuals younger than the age of 30, have HBs antigen positive. So this is a great success of the immunological approach of vaccination. The second success is, of course, the patient's own immune system, because if you look at hepatitis B infections, 90 to 95% of adults who get exposed to the virus clear the infection spontaneously. So really showing that the immune system itself is highly capable of clearing the virus, but not in all patients. So in the minority of patients, this is not the case. And so what I will show you is two topics. I will discuss the mechanisms that cause persistence, just to give you an idea of what immunologically is going on. And the second thing is to show you a little bit of new insights on the HBV clinical phases, because there's a lot of confusion about the immunology of hepatitis B and a lot of things that are still unknown. Well, first of all, a brief introduction. So there are four major compounds in an immune response to antiviral infections. One is the interferon system. Interferons are very active in inducing a lot of proteins that have antiviral activity within the infected cell. The second one, you're well aware, B cells. B cells, by their ability to produce antibodies, are able to neutralize viruses in the circulation, also have other functions, but neutralization is an important point. The third one is natural killer cells, but also CD8 positive T cells. They are able to really kill infected cells by the release of proteolytic enzymes and various other enzymes. So in a direct, in a specific or a non-specific way, they can recognize an infected cell and kill the cell. So those are the four pillars of the immune system that are able to be involved in an antiviral response. And when you look at the kinetics of these responses, then overall what you see is very early in the infection, very high interferon responses, followed by NK cell responses, and that already in the case, for instance, of a resolving infection, a rhinovirus infection or an influenza virus infection, you get a reduction of your viral load, and that is then followed by T cells, B cells that clear the virus, etc. So that is an effective immune response against an acutely resolving virus, but that doesn't happen in the case of hepatitis B. Because hepatitis B is very efficient in avoiding recognition. It's very efficient in doing that. It also is very efficient in avoiding cell damage during the replication phase of the infection. So there's very little cell damage. And also, thirdly, it secretes viral proteins, HBs, HBe, that modulates innate immune responses. And the consequence of that is that you have very low activation of your innate immune response. Very few cytokines are produced, and most of the time in the early phases of the infection, patients are asymptomatic. So it's a very weak immune response initially. A lot of people talk about stealth viruses, but it could also very well be that there is active suppression and just not recognition of the virus itself in the early phases. But what happens in the situation where the patient develops into chronicity? So initially, if patients have T cell responses, they're detectable, but if patients develop towards a chronic infection, these T cells most of the time collapse. And as you can see here with the green lines, during the development of chronicity, the CD8 cells reduce in numbers and you don't see them too much anymore. And so if you compare a chronic versus a spontaneous resolution of the infection, then what you can appreciate here is that when you look for CD8 positive T cells or CD4 positive T cells, then overall you cannot find them in the blood in a chronic infection. So they're hardly there. They're not in the blood, but they are present in the liver. So what you find here is, what you see here is that in the liver, variable amounts of HBV-specific T cells are present. In the blood and the liver, in liver but not in blood. And you see that when you have very high loads of HBV DNA, that you have lower numbers of HBV-specific CD8 cells. So there's something going on with the amount of virus and the amount of T cells. However, if you look at biohistology in the liver, then what you can also see that around the portal tract areas, you find very high amounts of HBV-specific or of T cells around the portal tract areas in the liver of infected, chronically infected HBV patients. So the cells are there, but you hardly have any damage. So you hardly have any damage in the liver, despite the fact that there are a lot of T cells around. And what happens in these livers is there's a lot of negative regulation going on. So what you can appreciate here is if you look at, for instance, Kupffer cells, they are able to suppress the activity of the CD8 cells. Also natural killer cells that are present in the liver are also able to suppress the activity of your CD8 T cells, and you have a lot of regulatory T cells, myeloid-derived suppressor cells. So you have a lot of cells that are all that their activity is all focused on inhibition of the activity of these hepatitis B specific T cells. So the environment in the liver is, in that sense, immunosuppressive, but also because you have a very high antigen load. There's a lot of virus in an HBV infected, in a chronic HBV infected patients. That very high antigen load also makes that the immune system is more or less switched off. And especially the very high doses of virus, the very high doses of antigen, are very well known to inhibit T cell responses. Because what has been shown, that is if you increase the amount of antigen, so the more antigen you have, the more activities of T cells are at a certain point switched off. So you first lose, in this case, your cytotoxic ability and your IL-2 production, and the more antigen is around, the more activities at a certain point of your T cell you lose. And that is called an exhausted T cell. And exhausted T cells are characterized by the presence of certain molecules on their surface. And one of the most well-known molecules is PD-1. And what you can appreciate from this slide is that if you look at hepatitis B-specific T cells in the liver, as well as in blood, then these cells have a very high expression of this inhibitory molecule PD-1. It's higher on HBV-specific T cells than on other T cells, so it's higher on HBV-specific T cells than on CMV or influenza-specific T cells. It's not only PD-1, it's a lot of different molecules, CTLA-4, LAG-3, etc., BTLA-4, TIM-3, those are all molecules that are expressed on this exhausted T cell in the hepatitis B situation. And the question, of course, is this reversible? So is this T cell that you find in the chronic HBV patient, is that forever completely functionally impaired? Or can you revive that? And so one of the logical experiments then to do, and that has been done by Calavarari's group in Italy, is he simply treated patients with nucleoside analogs, tenofovir and tenofovir, and found that, and that is in the lower graph, that indeed you can restore these T cell responses in chronic hepatitis B patients when you successfully treat with nukes. Also, it has been shown when you specifically, very specifically, block, and this is done in cell cultures, but block the activity of PD-1 by using antibodies, then you can indeed enhance the activity of your hepatitis B-specific T cells in this situation. And at the moment, these molecules are also of these antibodies are also in clinical trials. They're in clinical trials for the treatment of HCC, but because of a lot of HCCs or HBV induced, the efficacy results will also be available on the effect of restoration of your immune system to HBV. So besides these effects on exhaustion markers, so the exhaustion that there is functional impairment, there's also very active inhibition by cytokines and also indeed inhibition most likely by the presence of very high amounts of antigen. And so there are high, as you well know, there are very high levels of HBs, HBe antigens around. We have no idea what the consequence of these very high levels of viral proteins are on the immune system. So it could very well be that those very high levels also affect the immune system and negatively regulate the immune system. So the biggest problem in Chronic viral infections like hepatitis B is that the CD8 positive T cells, but also some other cells, are functionally impaired. The restoration of these T cells may enhance your immune system and maybe have as a result that you clear the virus. But of course, there's also a downside, because if you have a very strong T cell response directed against the virus, then you will also induce a lot of pathology, a lot of damage in the infected liver. So it is a balance, and maybe because you have indeed these very weak T cell responses around, maybe that makes that the damage that is induced in the liver is very mild. There's very mild pathology, and that is probably the reason why you have this state of chronic infection. So I would like to change gears a little bit, and we'll also tell you a little bit on the HBV clinical phases. Also because after the talk of Heiner, he made quite clear that indeed a typical hepatitis B patient is not a very homogeneous group of individuals, but the disease is quite heterogeneous. And so you're well aware of the 4 phases that are defined in the clinic and that serve as a guideline for the treatment of patients: the immunotolerant, immunoactive, inactive carriers, and HBe negative hepatitis, in which only the situations where there is ALT increases, fluctuating ALT, are strong indications to treat patients with the various compounds. Well, these categorization of the 4 clinical phases are, as you're well aware, they are based on the presence or absence of HBe antigen, but also the levels of ALT and the viral load. Very strict definitions. And what we wanted to do is to very immunologically try to characterize these 4 phases and try to figure out what really is the difference between these 4 phases and what does cause these fluctuations in viral load and ALT. Is it an immune phenomenon, or is it maybe the virus that causes these fluctuations in viral load and ALT? And if you look in literature and what the general hypothesis is, then the general idea was that during the immunotolerant phase of the infection, the immune system hardly plays a role, and it's mostly the virus. So the virus is there, the virus replicates, there's hardly an involvement of CD8 positive T cells or other immune cells, and that is the reason why it was called the immunotolerant phase, because the immune system more or less tolerized the virus. In the immune clearance phase, it was more or less equal. There was some sort of balance between viral replication and the immune system. And when there was remission, then the immune system took over and the virus was less important. So purely based on this, the terminology was designed, and that, from an immunological point of view, always caused a lot of confusion, because immunologists always consider immune tolerance something completely different than how clinicians see it. So what we did is we did a very broad study. So we put our complete toolbox in this study, and we tried from a lot of different approaches, tried to get an idea of what really was going on in the clinical phases of patients by selecting very strictly a group of patients that fitted perfectly in these various categories. So we looked at our outpatient clinic and selected the patients that perfectly fitted in these categories by also selecting patients with no comorbidities, patients with hardly any fibrosis, etc. And by doing so, we assessed them immunologically. And first of all, a very simple test is we just did the differential analysis. But if you look at these patients, the number of granulocytes, monocytes, leukocytes, everything is completely similar when you compare these 4 different groups. So despite the fact that they had completely different ALT levels, viral loads, etc. And we also did a very detailed immunology of a lot of markers, activation markers, etc., NK cells, dendritic cells, CD4, CD8, B cells, etc. We also found absolutely no differences in frequencies of these populations. Well, then nowadays the technique to really study very broadly what is going on in immune response is to do gene expression analysis and to do RNA sequencing and those kind of fancy techniques. So we did that on blood of these patients, and we grouped then all the genes that we found into certain functional categories. So we grouped all the genes that had anything to do with B cells, NK cells, etc. And if we did that, what we found is that when we looked at the 4 different phases, then when we look at the immune tolerant phase, we found that there was a very high activity of interferon stimulated genes, whereas when we looked at the genes with fluctuating ALT, we found a strong activity of genes, anything to do with natural killer cells or cytotoxicity. And also we found that after the immunotolerant phase, the B cell component came up. So this at least gave us some idea of what really was going on in these different phases and what the role and the importance of the immune system is in causing this fluctuation. We did it in blood, but we also did it in the liver. In the liver, we found more or less the same. We also found, again, an importance for B cells and NK cells in the same phases. But we also found, and this is, if you just look at the colors, we found a lot of differences in a lot of genes in these various different phases, in these 4 different phases. So really showing that the immune system is important and that the immune system could very well determine what the 4 clinical phases is, besides probably an involvement of the virology as well. When we did immunohistology for T cells, then what you can appreciate here is that the immune tolerant and the inactive carrier phase had very low numbers of T cells, and the other 2 phases had high numbers of B cells. And when you look at the B cell immunohistochemistry, then you see, especially in the immunoactive phase, that we find a lot of activity for the B cells, and that was also what came out of our gene expression analysis. So I think this data shows, and I think this is really one of the first studies to really systematically try to figure out what is really going on in these clinical phases, is to show that the immune activity is really different between these 4 clinical phases, and that we have to appreciate probably also a role for B cells and interferon stimulated genes in causing these different phases. And that has been that is a cell type at least that has been completely ignored in the whole immunology of hepatitis B infections. And thirdly, NK cells or cytolytic activity we found to be very important, and especially those phases that are fluctuating ALT levels. And so that is quite important, because also we have no idea what really causes this fluctuation. And again, if it's the virus or if it's the immunology that causes it. Well, one of the things that really needs to be done, and that Heiner also alluded to that a little bit, we have a lot of variables in hepatitis B infection. So not all patients are the same. We have different genotypes, different genders, different ethnicities. There's a lot of different causes or co-infections, etc., that may influence your chronic infection. And I think it's really important to now that we really start to figure out what is happening in these patients and what the various variables is, because clinically indeed you have this observation that NUCs behave differently in the HBe antigen positive and negative patients when you look at the numbers of patients that get S loss. We have absolutely no idea what causes it. We have absolutely no idea what causes or what the effects is of HBs in the circulation of patients, what the effect is on the immune system. But we also have very limited ideas of what the effects are of co-infections on an infection with chronic hepatitis B. So I think to really start to figure out from an immunological point of view what is really happening in these patients, in this very heterogeneous group of patients, is really important also for the design of the novel antiviral therapies. Thank you very much. Thank you.